Hello, my name is Rebecca Rooney, and I'm an undergraduate student studying applied ecology and environmental science at Michigan Technological University. Today I'll be presenting my senior thesis project examining plasticity of Japanese beech across its native range by looking at changes in leaf anatomy across the vertical length of the crown. Multiple factors affect changes in leaf development within a single tree crown. For example, xylem tension and water-related stress increase from the bottom to the top of tree canopies. Also, external environmental conditions such as light, wind, humidity, and temperature vary along this gradient, all of which can affect leaf development. Together, these changes in leaf anatomy, which develop under specific microclimate conditions, can help predict acclimation potential of a species under climate change. High plasticity allows individuals to better cope with environmental change. This is critical for all tree species as they must exist in a single location for their entire lives. High plasticity is even more critical for species that are long-lived and slow-migrating, like Japanese beech. Our first hypothesis in this study was that the primary mechanism controlling leaf development would be light availability. Second, we hypothesized that plasticity would be strongly influenced by latitude, with high plasticity trees occurring at the northernmost regions of Japanese beech's range. 5 sites were selected across the species range. Leaves were sampled from 1 to 2 trees at each site from 5 height classes. Leaves were then thin-sectioned with a freezing microtome, photographed, and processed in image J to measure leaf thickness, palisade mesophyll cell thickness, percent intercellular airspace, and epidermal cell area. We estimated intra-crown leaf plasticity using canopy openness and height as covariates. Canopy openness is a proxy for light availability, and height is a proxy for xylem water tension. Plasticity was quantified as the slopes of the regression lines of the leaf anatomical variable versus both height and canopy openness. A second plasticity index was calculated as the difference between the maximum and minimum leaf trait values divided by the maximum value to create a scale from 0 to 1, with 0 representing no plasticity and 1 indicating very high plasticity. These plasticity index values were compared with climatic variables across the species range. Our results show that traits vary across the vertical length of the crown and are more strongly influenced by different mechanistic factors. Leaf and palisade thickness and epidermal cell area increased from the lower to upper crown and were all more strongly controlled by light availability. Intercellular airspace decreased with height and was more strongly related to xylem water stress. We found both indices of plasticity to be greatest at sites that were lower in elevation and that had greater mean annual temperatures. So what does this tell us about the factors influencing plasticity and its role in the future of Japanese beech? First, the mechanisms directly influencing leaf anatomy of the species are generally more controlled by light. Second, populations of Japanese beech located at higher altitudes and in areas with lower mean annual temperatures may be more vulnerable to climate change as they had the lowest plasticity. This decline may be most noticeable at the southernmost reaches of its range, where populations are already isolated at high elevations. There are already models predicting a large decline of Japanese beech by the year 2100, with some 9,300 square kilometers of suitable habitat being lost by this time. In developing a more comprehensive understanding of regions where this species will be more resilient, it is possible to improve our management and conservation efforts for Japanese beech.